Okay. All right. Let's see. Just change something here. Can you all see my screen?
Yes.
Yeah.
It's just a little wonky for me. Okay, there we go. Okay. So yeah, last week we convened a meeting with patients, and the reason we did it was we've noticed that we're about 10 patients have showed up over the course of the past several weeks. And so we wanted to have sort of a patient-only meeting with them to do a little bit of market research, kind of understand what their needs are, what they want on their healthcare journey. Mike Yancey, who's on today, was one of the participants, and we had 3 others. It was about an hour and a half meeting, and it was really just to kind of get to know them, first off, understand their journeys, and then really just understand, if you had one thing that you could change with your healthcare, what would that be? And there were 4 themes that sort of emerged. The first, which I thought was really interesting, was from Steve Abbott regarding the need for improved doctor-patient communication. So he actually is starting an initiative. He is the founder of the Cincinnati Cancer Foundation. I'm not sure I have that exactly right.
Not founder, but executive director or some title.
Okay, thanks, Brad. And so he understands that there is a huge need to try to improve what we would just call doctor ease. There's so much terminology that is thrown at patients, and it's difficult for patients, advanced prostate cancer patients in particular, who are, given this diagnosis and have to come up the curve so quickly. There's just a whole new language that patients have to learn. And so he has an initiative that he's trying to drive, and he wants our help in doing that. And so that's the first theme. The second theme was from Ken Anderson. And Ken and I have known each other for a while. He's a member of ANCAN, and he really sees a lot of value in what we're doing, because this initiative, the Prostate Cancer Lab, is really focused on better diagnostics. So he wants to be able to, he recognizes that every patient is different. So like Rick and I, we both have prostate cancer, but genomically we're completely different. And so we want to, have more data-driven decision-making, predictive response, treatment options. So a lot of what we've already talked about. So he's very aligned with our mission. The third area was around having a strategic partner. So Mike, hopefully I'm getting this right. Mike really needs somebody who is going to be alongside him. He has sort of really been bounced around from a number of different doctors. Being in a rural setting, I think, has been challenging for him. And luckily, he's been able to get care at MD Anderson in Houston, which is, I think, changed how his care is being delivered. But still, he's many miles away, and he needs somebody who's really going to be alongside him. And that kind of taken a part of his discussion into a fourth theme, which is that we need quality care regardless of location. It's very clear that rural settings lag metropolitan areas. And I know that this is something that is on the agenda for the Prostate Cancer Foundation. And now I think we've got some information that we can bring to them, which is, it's anecdotal, obviously, but it just reaffirms that as the Prostate Cancer Foundation wants to raise the water level of medical knowledge across its doctor network, certainly how it addresses what happens with rural settings is very, very important. And Mike's a great example of that, being in northeastern Oklahoma, where he has fewer options than, say, Rick or myself, being on the West Coast in San Diego and in the Los Angeles area. So, it was a great meeting. We talked about how often we want to meet. There's going to be a little bit more discussion about that, but what we're thinking right now is that we're actually going to put together like a patient board, and probably formalize this where we're going to meet on a monthly basis. And then we'll take information from those discussions and feed them into this format as well. Any questions?
Brian, this is John. I had some, I guess a clarifying question, but some input, hopefully helpful. On strategic partner doctors, I kind of heard my listening for that was two different possibilities. One is a physician who might be a treating physician or an integrative oncologist or an integrative naturopath who specializes in oncologist. That's a kind of a category of strategic partner doctors. And then the other one is the role that I and other doctors play, which is the medical advocate role, which is kind of like having an independent consultant, and I'm looking over everything in the case. You know, is the diagnosis accurate? Do we need to send it to a specialist who can overturn the diagnosis or make it more accurate? Who are the oncologists that would be open to, you know, expanding the range of options? Who's in your area you might not know about? Those kind of things. New testing options, like I suggested earlier to Rick. That's a strategic partner. And so there's a whole world of advocacy that you guys probably know more about than I do, but there are a group of medical doctors that do medical advocacy that the mainstream I know about, which is where I was trained by Mark Renneker, who kind of founded the field. But there are,
I know.
nurse.
Sorry,
You know,
sorry. He's a Mavericks charger. He's a he's a big surfer. That's how I knew about him. Yeah,
exactly. Yeah.
Yeah.
When his office calls the patient and tells the patient, you know, we have to reschedule because something's come up. All the patients know the waves have come up, and Mark was out on his surfboard.
Right.
They're also thinking of Laura Poole, who is a advocate, and you could say she's medical, but she's not a doctor. She's a nurse, but she had 30 years in oncology care and is research driven and works for Commonweal, the Commonweal group up in Marin, who gives super competent kind of strategic ideas. Kind of medical doctors, I think we maybe step in a little bit more. She said she's more likely to say, here are the resources, look into these. But so anyway, those are two listenings I have for when you say strategic partner doctors.
Yeah, that's super helpful. You know, there's a few different ways to interpret it, and Mike can certainly add some color to this too. But and thank you for adding more context and clarity. There's also this element of right now, so many patients only get 20, 30 minutes with their doctor. And particularly when it's complex, 20 to 30 minutes just isn't enough. And so, you know, that's why I put in this quote, you know, be alongside me on my journey. That's what we need. And unfortunately, so many patients don't have access to some of the resources that you just mentioned. And so maybe there's an opportunity to figure out how to scale that or change the model a little bit.
Yeah, and related to your improved doctor-patient communication, sometimes my advocacy work, I'm prepping a patient for a conversation with their oncologist. And you know, this is what, because I'm trying to, not to make about me, but my model is to empower the patient. So how the patient can have an informed, empowered, effective conversation with the oncologist. Sometimes it's what we might spend, you know, 45 minutes on. Usually doesn't take that long, but you know, half hour to hear the questions, and because it takes, like you said, it takes patients a while to be able to understand what's going on and to make effective requests.
No, that's great, and it's a really important need and service. So thank you.
I just want to add something here. If I may. Can you hear me?
Yes.
Yes.
Yes, thank you. And as I said, I've been working with this personalized data, mostly omics, for many years. And number one issue I see is the problem of the design of those experiments. The reason I'm saying this one, I'm seeing the predictive response here, which is very important because majority of those experiments, they start with a fuzzy question, and they just try to explore. It's about exploration. It's not about prediction. They don't start with a question and try to answer it. They say, let's go and find if we can see something new. Mm-hmm. Exploration generates thousands of new questions. And then we go through this process again and again, and it's going to be very unsuccessful. This is very important. Based on my experience, it's good to start with the question. For this patients, what is the most important question? Then see how we can answer that question. Can we answer it with the existing research, or we need to do something new? Then the idea of predictive research is very important because it's about answering a specific question. How much time do I have? Is it going to be positive? This treatment is good, or is for me, or it's not? Then this is important. Question-based predictive approach instead of just go and explore and see what we can find.
Yeah. Yeah, I think we're going to talk a little bit about some amazing work that Rick Stanton has done on NCCN guidelines. And Saeed, we had a really good conversation about that, about how you, for example, integrate diagnostics, testing to, for example, eliminate certain treatments that just wouldn't benefit a patient. For example, if you understanding, your hormone sensitivity, for example, there's an ARV7 test for that, right? There are potentially organoid models that could help us understand whether or not a patient is going to respond to chemotherapy. So there are others, right? And that's what we're trying to get to, I think.
Yeah. If I could just jump in as moderator, I think two things. One, I think what Dr. Laird was saying is, there are different kinds of people that can help with the patient, getting them educated and being able to ask the right questions. I think what Saeed is saying, just generally, not specifically, but in general, it's helping patients ask the right questions, and that's what would be entailed in having a good conversation. Nice to see Emma Stibelman here. Thank you for joining us. Thank you for taking the time. But I think we should turn it over to Allie. Allie's not feeling well today, so I appreciate that she showed up in spite of that. It's also the case that because we've lost 20-25 minutes of our hour, that I'm sure we won't get through everything in the time we have remaining today that we could have with Allie. But that was intentional. We expected that we might need to have a second meeting to follow up with some of the things I'm sure will come out of this conversation. So it's not a failure. We'll stop promptly at the hour, but it's not a failure if we don't get to everything today with Allie. So with that,
yeah, thanks, sir.
Yeah, I thought Brian was going to set it up for you, Allie, if you don't mind.
Yeah. No, that'd be great.
Yeah, if he could just do his since you're going to be talking about his case, he'll do his medical history and kind of the questions that he's confronting.
Okay, great. Can everybody see my screen? Okay, cool. So I'm going to be very brief because I really want to make sure that Allie has as much time as possible. But very briefly, I was diagnosed in 2016, had a prostatectomy. Unfortunately, it came back, and then I went on this wild ride. So went on first line hormone therapy with Lupron, did radiation, and then saw an uptick in my PSA. Then I went to a second-line hormone therapy with apalutamide, saw an immediate response to that, which was great. Ended up taking a little bit of a holiday in December of 2019. You can see step 5 there. And unfortunately, you know, when I had biochemical recurrence at that juncture, but it became visible as soon as I went off of all hormone therapy. And in August of 2020, point number 6 on this graph, you know, we discovered that I had 6 metastatic lesions in my peritoneum. We did surgery, and then I started a combination of chemo and pembro. You can see my PSA began to drop through all of that. I took 6 rounds of chemo, stayed on pembro for about 12 months, and then began to see a bit of an uptick in the September of last year, and did some more imaging and found that I had 3 metastatic lesions in the peritoneum. Prior to that, you know, there was no visible mets after the surgery and after chemo. So at that juncture, I am now on abiraterone, and you can see I've had a PSA decline that's gone from 0.91 to 0.45. So that's my quick history, and I am going to turn it over to Ali.
I'm sorry, just quick question. What was the Gleason score? Do you have that one?
Yes, I do. It's a 4.3 with tertiary grade 5.
Thank you.
It's the grade 5. That's the bad stuff, obviously.
Thank you.
Okay, Ali, I'm going to let you. Let's see.
Do I have the presenter rights? I think I do. Let me try to share.
Actually,
this.
Brad, you. Yeah, there you go.
You see it?
Yes.
Yes.
Okay, thank you. Thanks for having me. I hope what I present will be helpful. I spoke to Brian. We met last year virtually, and I know there's been some history of getting CureMatch reports before, and I can speak to the past report, but keep in mind that that was, I think, 2017. Different data and, of course, different knowledge was out there in the world. And what we do, I want to explain really briefly. You can think of it as knowledge representation and reasoning, as opposed to machine learning type of AI. And it's really important because we're not trying to make a prediction. It's more like scoring a molecular fitness of therapies, drugs, to the markers of molecular type in each individual profile, which as our founder and chief medical advisor, Dr. Kurzrock, says, is like a snowflake. Everybody's cancer is unique. Everybody's cancer is a snowflake. So the idea of this CureMatch brainchild of hers that was born in 2015 was to be able to emulate sort of the tumor board type of reasoning with all the knowledge at hand, so that for any unique new case, even if some mutations have never been seen before in the arrangement that they align in a given patient, no matter how unique a case is, if there is a way to therapeutically address or drug the target, so to speak, then the match will be considered by our system. So I'm gonna just tell you how it fits in sort of this whole therapy matching space, and then we'll go straight to the reports. So in what we do, we basically don't attempt to call variants, markers, what is pathogenic and what is not. We're not trying to assign the initial clinical interpretation and variant curation and significance the way that labs do. So we actually go next in line after NGS reports are received, and not just the NGS reports, but also molecular data on proteomic level, gene expression level from other types of reports can be integratively analyzed. So we come in after the NGS reports, and unlike some of the companies that do a really beautiful job of also showing what are the known therapies associated with markers that are known as actionable or pathogenic, we also cover novel combinations. So when we think about the 300 or so available cancer drugs, that's what we work with to consider all the millions of ways to mix and match those 300 drugs into 3-drug or 2-drug combos, whether they're known or not known. And customized combinations, as you know, is something that a lot of tumor boards end up concluding with, is recommendations to take certain drugs that are available, but combine them in ways that may not have been previously evaluated in clinical trials or approved. But the drugs themselves are basically already available. And that, in our experience, covers, the novel combinations cover about 99% of the top molecular matches for each patient. So in our system, we basically
Ali,
take
can I
care of
just
this
interrupt?
complexity.
Ali, can you just go back to that? Just define some terms and some things that I know are obvious to you, but that might not be obvious to everyone. NGS stands for next generation sequencing, and they're typically looking at an oncopanel of how many genes?
Oh, we are not personally limited by what type of lab or panel is used. Of course, larger panel and known, more reliable sort of clinical CLIA level lab tests are probably more favorably welcome. But we will take any sample type, whether it's liquid biopsy or solid tissue, any company, any lab that does NGS analysis, whether they interpret the results for therapies or not, we will take that and take the markers that are discovered and we'll CureMatch it, so to speak. So taking the molecular profile, whether it's DNA level and or having RNA and protein alterations as well. And then that arrangement is unique for each patient, and we will CureMatch it, meaning we will come up with therapeutic matches based on molecular ways of targeting this molecular profile.
Okay, so just again, just kind of get a sense of scale. I'm just trying to get a sense of scale. So the number of genes that one of these companies looks at, like I think a Foundation Medicine looks at 200, 300 genes, and that's what they're providing input. And then they'll identify within that, they'll say, we see variants in, and what is it, typically a half a dozen, a dozen? How many biomarkers are they identifying that might be useful and therefore would lead to your analysis and be treatable?
Great question. So usually what we see, it's about 5 or 6 markers on average. And this could be in a way a factor of our reality, is that when patients turn to CureMatch, when doctors want to order it, is when there's something that's really not trivial and it's difficult to envision out of all these millions of possible combinations, which 2 or which 3 drugs to really put together. And that's where we come in. And unfortunately, this is used often too late when the cancer is advanced or the problem is complex. And we're hoping that, first of all, NGS will be ordered a lot more routinely earlier on in the patient's journey. And then everybody who gets NGS results will also qualify for being CureMatched as almost, standard of care. But right now, our reality is such that we get quite complex cases, and we see anywhere between 2 to sometimes 15 or even 20 markers, especially if you have expression data, RNA gene expression data with differentially expressed genes, that adds markers. And on average, we see 5 or 6 or so markers that are of any kind of significance.
Thank you.
Ali, this is Rick. Could you go back a couple slides? Sorry, back to, I guess, 8.
I was showing this one. Is this one or the other one?
Yeah, that one. So known therapies, in the bottom, we see in teal less than 1% of matches. I'm not sure. let me say what I think that might mean, but please correct me. So let's say I have a mutation that has a known therapy, which is kind of rare. Is that what the teal means? Like 1% of the time you actually have a mutation that's directly targetable. Is that what that 1% means? And then the other 99%, like you have these mutations that, you need the CureMatch system to really, it's not so obvious. Is that how I'm interpreting that bottom bar graph?
You're pretty much on the right track. I just wanted to add to that. So it's not just per mutation, like drug mutation one-on-one. It's more about out of the therapeutic matches, which we, as you'll see on the reports, we present on basically the top page. Out of the top therapeutic matches, out of all of the reports that we've done, what we see is only much fewer than 1% of the time, they end up being kind of ready to go available combinations or monotherapies that simply well address the whole molecular profile, not just one mutation, but the matches to the overall profile. Like out of everything that's available, how many times is a given known available combination, for instance, targeting all 5 of these pathogenic markers, for instance? That's much fewer than 1%, which means, well, you need to take the drugs that are available out of those 300 and then mix and match them in customized, individualized ways that are still molecularly precise and justifiable. And that's what we do. So hopefully that makes sense what the greater than 99 and less than 1% is talking about.
Okay, we have a couple of questions. Jeff Waldron.
Yeah, might be a dumb question, Allie. How are you? I hope you feel better.
Hi, Jeff.
How are you? I hope you feel better. So,
okay, thanks.
This is probably a dumb question. So does CureMatch, I should know this, does it also incorporate drugs that are investigational? In other words, either drugs that are under clinical trial or under maybe available to patients under expanded access compassionate use programs because they don't have any more standard of care options. So does a combination sometimes include investigational therapies?
I love the questions I'm getting. Really, it's such a joy to talk to you guys. This is actually something that came up in my correspondence with Brian because there was a drug mentioned that's in clinical trials. And I'm so glad you asked because it's very important to mention that we just work with cure matching the available drugs, meaning if it's a completely novel investigational compound, it's not in our system. Only when it gets approved for at least some indication, even if it's non-cancer indication, but it gets approved for something, then we can use it.
Yeah, that's unfortunate because I've been working a lot with expanded access programs whereby, because the problem that Brian just described is you get in a trial and you may not get the therapy if you're getting the other side of the trial, so a randomized trial. So whereas an expanded access is individual approval to the pharma company and the FDA, but you can guarantee that if you're approved, you'll get the therapeutic option of an investigational medicine.
Yeah. So here's the thing. So far, our clients, and we work through physicians primarily, but also a lot of times it's initiated by patients, have not been turning to us specifically for clinical trial matching when there's investigational drugs. And historically, again, this is just the state of the art right now and the field. Historically, it's been such that when physicians turn to us, they know that they're going to be trying something different. And the case is very complex and often advanced to where they don't know if their patient will even qualify for a clinical trial. And so when we do present clinical trial information, it's more like to show you that the combination that shows up in our report is being given to humans. It's more like if there is anything, it's like evidence to show that, you know what, this combination, even though it's a customized combination and maybe not completely on label or just one of the drugs is on label, guess what? It's being trialed as such in these clinical trials. But we're not, so far, we haven't had a lot of demand for doing clinical trial matching for novel compounds. We basically take care of something that only we do so far in the field, is this complexity, going from envisioning millions of possible combinations, and out of those figuring out which ones are relevant for the given case. This is from a different report, just breaking it down, and then filtering, scoring, and ranking those to see which 2 or 3 drug combinations are best fitted molecularly for targeting the overall profile. And we kind of just focus on that, and then we're probably going to, you know, expand to other areas as well and include more things in our knowledge base and our algorithms. The framework is there, but we're focused and we're a small startup, so this is what we've been doing.
Thank you, but I think you and I are going to keep talking about this.
Yeah, Jeff asked about 90% of the question I was going to ask, but the other piece of that, and you touched on this, you're also looking at non-anti-cancer agents that are FDA approved as well, right?
Yes.
Okay, well, that's good.
There are quite a few, relatively few, of course, compared to the cancer drugs in our knowledge base, but there are some good examples of repositioning of drugs that have been originally approved for something else, but yet they successfully target some cancer targets. And when the molecular triggers are there, they will be considered by the algorithm. They may or may not score highest, but they will be considered scored and presented if they are a match. Yes.
Well, that's good.
Dr. John Laird, you had a question?
Yes. Hi, Ali. Thank you so much for making your presentation. My question is very basic. When you say that you identified typically, 5 or 6 pathogenic markers, and I assume that's the same on your chart here as actionable markers. I'm wondering what that means, because not every genetic mutation is pathogenic, and to be chasing I'm not an expert in this field at all, but to be chasing mutations that are irrelevant to the progression of the disease would be a concern for me. So how do we sort that out?
Yes, so you're absolutely right. And sometimes we just don't have enough markers to match, in which case we won't even produce a report. I actually participated in several of these hackathons, and there was one when there just weren't enough markers to produce a report at that stage. Then later on, something else was presented. But if there is a mutation and there is no pharmacological agent out there, and we can't invent one, then we're not there to propose something completely novel. This is not drug discovery. So what we'll do is, if there are markers that are actionable, that would be part of the 5. But there can be, absolutely, and we see that, pathogenic markers that are not actionable. And when we talk about Brian's case, the fusion is not considered actionable. There has to be really good clinical evidence for us to put it as actionable, for the linkage to drugs from a drug target to be made. And so right now the fusion is not actionable, but there are other markers that are actionable from different levels, DNA and RNA. And so that actually goes into the algorithm. I'm just going to switch from this to Brian's report in 1 second, but just not to be confused, this was breaking it down on the slide, but this is not Brian's report. This is a different example, but there were 5 actionable markers, but I believe in this case, there were 6 or 7 pathogenic or just clinically significant markers that were reported from NGS results. And again, unless clinical team overrides in such a way that they say, consider this pathogenic or don't consider that, we will take whatever the lab reports and GS pathology reports say, and we will CureMatch that. We don't reinterpret results. That's clear sort of territory. I hope that makes sense. Listen, folks, can I just quickly breeze through at least, the highlights of everything, and then take more questions before the hour is over? Is that okay? So look, all I need to say before I close this slide deck is that, and I just want to show the PDF report right away, is that this whole process of cure matching. So it gives you a score. It's called PreciMatch score. This score represents how well are you covering all of the pathogenic markers with the drugs that are available, that are presented in any given option. We show top 3 drug, 2 drug combinations, and also monotherapies, because physicians like to see that, and sometimes they do score quite well. So when we have something that is a therapeutic option that covers 100% of the markers of the molecular profile of the patient, perfectly, directly, 100% hands down covers it. The score would be, guess what? It would be 100%. If there is no molecular basis for matching, for instance, something can be on label, so approved in the given type of cancer, but there is no molecular trigger to any drug sensitivity, then the score will be 0. So it's not enough for us to have something that's, you know, on label that says this is standard of care. There has to be molecular justification or molecular indication for a drug. So this is a very important point I wanted to make. The scoring is purely on molecular basis, and then with all things equal, when something is on label, we will show this, and we will also show in our targeting description, you know, when it's on or off label. And then, so now with this molecular score, what we've seen time after time, just back to our practical reality, is that the higher the score, the higher we've seen, the better we've seen progression-free survival and overall survival outcomes. And the worse the score, the more we've seen of very unfortunate data set points. I don't even want to say how much this actually pains us to see the data set, because behind each data point, there's a life out there where the low score behind the therapy that was given to these patients actually resulted in very poor outcomes. So this is the, you know, I just want to make sure that everybody knows that this whole cure matching and scoring, it does have merit. And since I came to CureMatch, we've been lucky to collect even more evidence. And right now, I'm really proud to say that soon we will be coming out with a series of papers that show with the latest our algorithm and knowledge base with the latest outcomes, even on a bigger data set. The same pattern is true, and that's really important, that the molecular matching and the degree of molecular matching is really predictive of outcomes. So that's basically where we come in with the decision support or therapeutic intelligence decision support that we provide for physicians. Now let me open a different screen because I want to get to Brian's report. Okay. Does everyone see this page right now? Okay. So again, the 4.5 million, that doesn't change. If you just do the combinatorics of the 300 or so drugs, you will get this very easily. But this number changes. So the number of potentially relevant combinations that the engine considers, it's a way to mix and match any of the or all of the drugs in 2 or 3 drug combinations that match any of the markers in the profile. Here, there were 6 actionable markers, but actually on the second page, and this will be shared or maybe is already shared, so you can review more. There are more than 6 markers, but only 6 were actionable. And that plays into the results. If somebody has, let's say, if they had 20 markers, but you can only hit, for instance, 8 with combination of 3 drugs, then you're not going to get a score of 100% because you're at best hitting less than 50% of the markers. It's just as simple as that. How many birds can you shoot with a few stones that you have? Because only up to 3 drugs are routinely considered. Although Dr. Kurzrock will tell you about her and her colleagues using 4 drug combos and just starting at lower dosages, and there are all of these different strategies. Now, when we look at this report, the top 3 drug combos, then there's 2 drug combos, and then monotherapies, they do not address the marker that's the fusion, because we're not counting that as an actionable marker. Actually, in our early report, we did, but it was such an indirect connection that with time, it did not pass our curation standards. And so we do not consider it actually targetable, but all other markers from transcriptomic and genomic data were taken into account. And the way that they are addressed actually follows in all of the subsequent pages, where you can see things called targeting description. Please, when you go through reports, we're not going to go through all of this right now, and I just wanted to navigate you along. So when you go through targeting description, you can see what is it that is being targeted by this top 3 drug combo, by the second scoring 3 drug combo, and how directly or indirectly it's acting on, you know, the markers at hand. So sometimes the marker, it's not necessarily a drug target that we want to inhibit. It could be a tumor suppressor. It could be something that cannot be or should not be drugged directly, but it's a marker of sensitivity. In that case, we will also display it in targeting description. And that's where we'll show if something is off or on label. And very importantly, whenever possible, we have alternative drugs presented. For us to say something is an alternative drug, it's very a CureMatch specific thing. Not only do the drugs have to be of the same therapy type and the same targeting mechanism, they also have to have the same score. So when something scores, let's say, exactly the same as trametinib, and is the same therapy type and targets the same exact mechanism, only then will it be called alternative drugs. Only then it's seen as equivalent, because sometimes multi-kinase inhibitors, they can be alternatives for one patient in one case, but not alternatives of each other for a different patient. Depends on which of these targets are in the profile. So this is where you'll see alternative drug for apalutamide, alternative drug for olaparib. So different PARP inhibitors would be there. And then an alternative drug for trametinib, another MEK inhibitor. So the MEK inhibitor is there because BRAF showed up. BRAF showed up. We can scroll back up and see how this works. Right, okay, it was a transcriptomic variant. So this is a way to navigate through the report and see why certain combinations are scored, how the rationale works in terms of the targeting. And again, if there is a trial that is active that actually explores that same exact combination, it will be listed as also an example or listed under this table targeting description. So there's some under the 2 drug combinations. And then we move on to monotherapies. Something that you may not be very familiar with. I'm just going to make this one comment and then pause. We do show an option for indirect targeting of p53, which is unusual because p53, it's not something that can be directly targetable, but each time we show references to expose not only the reasoning of targeting, but also the references to show why that is. And that's because downstream of p53, there are hundreds of pathways, and we know that. But downstream of p53, there are angiogenesis pathways that are related to growth and proliferation that clinically have been shown to point at VEGF and VEGF receptor as potential targets. So meaning that patients that had p53 alterations, of all things downstream, when VEGF and VEGF receptor were targeted, those patients seem to show some clinical promise of that type of strategy. And so it's not necessarily just angiogenesis specific downstream of TP53. There are a lot of growth and proliferation programs, and you need to have the blood vessels also grow and proliferate in order to even recruit them and as part of the angiogenesis program as well. But VEGF and VEGF receptor, they're growth factors, growth factor receptors, which can have effect not only on endothelial cells, but also on cancer cells and promote proliferation. So that's the connection there, and we provide evidence, and you can see our references towards the end. So now I'm going to pause.
I think there'll be a number of questions. I know I have some, but why don't we let Brian, who's being analyzed here, ask the first round of questions. I know, Brian, you have several.
Yeah, so Ali, thanks so much. First off, I really appreciate all this incredible work. It's really tremendous. So, and I asked this question offline, but I'm going to ask it again because I do want to make sure that I understand it. So in 2017, we did this, and my highest prostate gene score was 70%. And in this case, it's now 32%. Can you just talk through, obviously the decline is not good, but can you just talk a little bit about how that, why there is that change?
Absolutely. So first of all, like I said, we had this flagged as actionable.
Yep.
And right now it's not.
Yep.
And if you think about it, this is a very rudimentary, simplistic way to oversimplify what our algorithm does. But let's think about it really, really simplistically. Let's say you have 2 markers and you address one of them. The score would be roughly 50%. If you have 20 markers and you address 5 of them, it's just going to be a quarter. That's as much as you can do given the picture of the cancer and all the pharmaceutical agents that are even available for us to CureMatch to address that picture of individuals' cancer. So one is this point right here. We're saying it's not actionable right now. And according to our standards, I think this is really a fair statement. And then there's more markers this time. So besides this being not druggable directly, now there's more markers this time than you had before. So even before, the way that this would have worked right now, if we just reran the same inputs as 2017, the scores would not have been 70%. They would be much, much lower. But right now, there were also other markers added, which give us options. It's not necessarily a bad thing, but out of all of the markers, there's only so many that you can target with any given combination. So for instance, here, we can address markers of sensitivity or direct drug targets as 1, 2, let's say 3, 4 at a time, but you cannot address all, I think it was 7 or so at once. So if you can only address, let's say 4 with a 3-drug combo at most, then you know that this is going to be kind of half of the picture that can be drugged at any one given time. Now, some doctors, like I said, even use 4-drug combos and just start at lower dosages and kind of titrate up. That's up to the physician to understand and decide on clinical appropriateness of it. But we match up to 3 drugs in our standard reports. So I hope that makes sense. We're just trying to cover as much ground as possible with as fewest drugs in a combination.
Okay, great. And then I saw your point about my question around CD276. So that's a clinical trial. So because that's a clinical trial, you're not integrating that into this report. However, first off, is that correct?
Yes, if it's something that I thought you mentioned a different marker.
Yeah, so you have in here CD274, but in the work that Rick and I did with Tempus, we identified CD276 as a marker that I express very highly for. And so I was curious if that didn't show up here.
I had the same question, sorry.
Okay, go ahead. Maybe I can stop.
Which is now a popular target for CAR T trials, very few though, and ADCs.
Yeah. So in our system, if it's something that's already approved and can be mixed and matched with other therapies in a combination, we would have included it. So for instance, if it was PD-1, PD-L1, PD-L2, the markers that are already part of the indication to, let's say, suggest pembrolizumab or analogous checkpoint inhibitors, then that would have been there. But because there was no expression reported to us, it didn't consider that. But let's say if you said, look, Ali, put into your system that there's PD-L1 expression and see what happens. I can already tell you that it would be in most of the, if not all of the combinations, because if there are markers of immunotherapy sensitivity, they are considered very highly by our algorithm. And that's for clinical reasons, because there could be more hope of a longer term progression-free survival, as you probably know. But right now, because it wasn't provided as such, I just took whatever was there in all the reports.
Well, I'm not sure if I worked with CureMatch a little bit before, and I'm very fond of the company, but I'm not sure in this particular case if the previous treatments are taken into account, because Brian already had pembrolizumab.
Right, but we didn't take it into account because there was no explicit sort of reason to tell me exclude this or count this. We have this as a physician's choice, where a physician can provide drugs to exclude, and then for whatever reason they may have, because they think that there could be contraindication because the patient is diabetic, or the other drugs they're taken, or previous history, like you just mentioned. That is when they say, match whatever you need to, but exclude the following. So that's when it's up to the physician or the inputs that are given to me to say exclude, do not score them. If there is no specific explicit reason to exclude it, then anything that will be cure matched, and it will score accordingly. In this case, there was no input to give immunotherapy in our top page. Again, it's up to clinical inputs. We don't say what is actionable and what's not. If you give it to me, and there's a reason for immunotherapy, it would match it. It would show up on the report. I would just have to rerun the report.
Is that okay, Emma?
That makes sense.
Yeah, that's understandable. The difference is that someone like me looks more for clinical trials rather than off-label treatment options. And when I look for clinical trials, I always have to keep in mind the previous treatments, the number and the nature of them. And CureMatch has this assumption that a physician will be able to prescribe off-label combination of 2 or 3 drugs.
Yep.
Which, as we know,
exactly.
happens rarely, unfortunately.
Yeah.
Even with the best justification. I know 2 patients who had recommendations from CureMatch, and their physicians worked with CureMatch and they received treatment and it worked great. But that's an exception in general.
I know. You're absolutely right.
Unfortunately, and it's very frustrating.
Right. Absolutely right. And that's our reality. We exist to provide this information to help inform decisions when off-label combinations are to be considered. Because if it's just for clinical trial matching, and I'm not minimizing that, it's just that there are other companies that do that quite well, but other companies will not look at, kind of like this tumor board style, right, when you're considering the drugs available. And then as soon as you have even 5 drugs available, to pick and choose which 2 to give or which 3 to give out of 5, not so trivial, because we also take into account toxicity, resistance, which combinations shouldn't be given, or which drug should not be given without another drug if the profile has this in it. It's very hard for any physician to keep on top of all of that. But I know we're not the majority case scenario right now, and we hope we will be. We're actually getting our CPT code later this year. We're in the process right now with the AMA, so we'll be more easily reimbursable. We're working towards it. We haven't had as much evidence as this year that's going to be even more published ever before. And we're in better shape right now. And I think that the field is really pivoting more than ever before. When I worked with Rick, by the way, at Human Longevity, and we just started the whole oncology program there, there were those more like progressive thinkers, and majority were really just not ready. And now I think the field is in a completely different shape than 2015, 2016, when our comprehensive cancer program was just kicking off at HLI. So it's happening. Hopefully it happens faster rather than slower.
I think that's a good place to wrap up. I'm conscious of the time. Brian, did you have any last questions or comments?
No, the only comment is, again, Allie, thank you so much. I really do appreciate it. Please extend my appreciation to the team. I know that there was a team behind this work, and so please share that with them as well. But really, really grateful. And, I'm going to have a conversation with Raina, about it and get her thoughts.
Thank you.
Thank you.